1 in 3 of us will get cancer at some point in our lifetime. This translates to about 2.5 billion people in this world who will get cancer at some point in their lifetime. Now, this is scary because this could be our family, our friends, or even ourselves. Cancer is a genetic disease that causes dysfunction of the cells. The human body has about 37 trillion cells in it, and these cells play a variety of roles in different parts of the body. But a single cell can cause cancer. Now, what is it about a cell that causes it to cause cancer and take over the whole body? Take a look at the human cell, for instance. It has about 46 DNA molecules, about 400,000 mRNA molecules, and about 40 million protein molecules. The cell represents an exquisite system that works as designed most of the time. But every once in a while, the cell can fail, and this sometimes results in cancer. Now, cells can fail in a variety of different ways, resulting in different types of cancer. If Tolstoy were a cell biologist, he would have said something like, healthy cells are all alike, but every cancerous cell is cancerous in its own way. So to truly understand cancer, we have to study individual cells. So how do we study a cell? In the last century, we put cells under a microscope, we stained them in different ways, and we were able to identify the phenotypes of many different types of cancer cells. But we couldn't look inside the cell and understand what's going on. More recently, with the advent of genetic technologies, we were able to study the contents of a cell in much more detail. We could identify individual variants in the DNA sequence that could result in cancer. Now, with single cell technologies like the Tapestri platform from Mission Bio, we have the best of both worlds. We are able to study cells in detail at scale. So we are able to get detailed information about every single cell in a cancer sample. This means that we can now study millions of genomes, transcriptomes, and proteomes. The Tapestri platform represents a resolution revolution. If you think of current technologies as being like an optical microscope, the Tapestri platform is like an electron microscope. It generates orders of magnitude more data that give you an unprecedented insight into the state of a cell at a given point in time. Of course, all this data now generates a whole new set of challenges. I'm Niranjan Vissa. I'm a director of software engineering at Mission Bio, and today I'm going to talk about how Mission Bio is partnering with AWS to drive a resolution revolution using single-cell genomics. So what is the Tapestri platform? It's a platform for multi-omics characterization at the single-cell level. Now, what does this mean? This means that we are able to simultaneously detect genotype and phenotype signals from single cells. Genotype signals are typically related to the DNA sequence of a cell, while phenotype signals are related to protein expression, among other things. With current technologies, to get the same type of data, you would have to do multiple experiments with different types of technologies and then laboriously combine data at the end to get a better picture of a cell than any single technology can provide. But now, with the Tapestri platform, you take the same sample, you run it through a simple experimental workflow, and then analyze the resulting data using an easy-to-use analysis tool to get a much more comprehensive picture of the cell. Now, this is the same workflow from a software perspective. The Tapestri platform supports targeted sequencing today. This means that every experiment must start with a panel. This panel typically contains targets that are genes related to the disease you are investigating. The primers designed by Tapestri Designer are then mixed with the cancer sample and processed using the Tapestri instrument. Within this instrument, we tag every cell in the sample with a unique barcode to help with later identification. And this processed sample is then put through a standard sequencing workflow, which results in FASTQ files coming out of the sequencer. These FASTQ files contain a lot of cell-level data, and this data is analyzed using the Tapestri pipeline application. And the resulting metrics are then summarized and visualized using a Tapestri Insights application. The focus of my talk today is going to be about the Tapestri pipeline application. So what is Tapestri pipeline? It is a web application to analyze data from the Tapestri platform. It performs a series of computationally or memory-intensive steps that really cannot be executed on your average laptop or desktop. We need the cloud to analyze this data. The workflow of Tapestri pipeline can be divided into 3 different steps. The first step is to first get data into the system. So this is the data ingestion step. And once you have data in the system, the next step is to process the data to get some useful information out of it. And the third step is to review these results in a way that makes sense to biologists. So let's talk about how AWS enables data ingestion in the Tapestri pipeline application. Now, the input data for Tapestri pipeline consists of FASTQ files. An analysis run requires at least one pair of FASTQ files. And each FASTQ file is typically tens of gigabytes in size. And some runs require multiple files of this size. So even in 2021, uploading large amounts of data to the internet is not an easy task. Further, our users are located in a variety of locations around the world, and they store the data in different systems, sometimes locally on their machines, sometimes in various types of storage setups like S3, for instance. So we came up with a variety of data ingestion methods that enable users to upload data in a manner that's convenient to them. Let's take a brief look at these data ingestion methods. The first method is a simple file upload method, where a user just drags and drops the FASTQ files into the browser. Now, this is a very, very simple approach that anybody can use. But the downside here is that the upload speed is limited by the user's bandwidth. This data ingestion method was built using AWS Cognito to ensure security and S3 transfer acceleration to enable the user to upload data to the nearest AWS data center. And this works well for users who have data stored locally. Some of our users have data that's already located in S3 in their own AWS accounts. Now, it doesn't make a whole lot of sense for these users to download the data to their local machine and then upload it into our application again. So we support direct import from S3. And the way this works is we allow users to integrate their AWS account with our AWS account and give our application permissions to access their S3 bucket. Now, this is typically restricted read-only permissions that allow us to only access the data that they specify. This is a much faster method of importing data, but it does involve some complexity in terms of AWS account integration. And we enable this via bucket policies that can be implemented using AWS Console, AWS CLI, or Terraform. Finally, some of our users store data in a third-party application called BaseSpace from Illumina. This, again, is a setup where users might have to download data to their local machine and then upload it again. So instead of doing all this, we provide direct integration with BaseSpace using API keys, where our application can import data directly from BaseSpace on the server side. And both importing from S3 and importing from BaseSpace are much faster than uploading from your local machine. Now, regardless of how data is ingested into the system, it always goes through a file validation step that runs an AWS batch before it is moved to a user-specific location in S3. And data is encrypted at all points. We use HTTPS everywhere during transit of data, and we encrypt data using KMS keys when stored in S3. Now, once we have data into the system, how do we process it? And how does AWS help with that? So the Tapestri pipeline application actually consists of multiple individual pipelines, each of which serves a specific purpose. For instance, we have a DNA pipeline that can be used for variant calling, and a DNA plus protein pipeline that can be used for variant and antibody analysis. Regardless of the pipeline, they all have the same steps. You first select the pipeline, then you select the input parameters, and finally you assign the FASTQ files to lanes if this is applicable to your case, and you click the submit button to start the run. Now let's work through a simple example. From a user perspective, let's say the user has a cancer sample that has been experimentally barcoded using the Tapestri instrument. And this sample is then analyzed is then put through a sequencing workflow that results in FASTQ files. And these FASTQ files are then uploaded into the Tapestri pipeline. So from an analysis perspective, the various steps that happen are first to demultiplex the reads and assign them to cells, and then map the reads to a reference genome and get rid of erroneous reads. Finally, for every cell and the reads associated to each cell, we run GATK, which is a popular variant calling application, to identify the variants that are present in that cell. Now, all these variant information is then summarized at a cell level into various output files, and this results in a single binary output file that contains cell level information. The format we use for the output file is HDF5. Now, what happens from an infrastructure perspective? How do we set up AWS to enable us to do this analysis? So in our current setup, when a job comes in, when the user clicks the submit button, we start a new EC2 instance. This is typically a Linux instance that has about 32 cores and 128 GB of RAM. And now you can see why the analysis wouldn't work on your average laptop. Now, once this instance is started, we copy the input FASTQ files to the instance, and we start the analysis run on this instance. The analysis typically takes several hours, and once the analysis is complete, we copy files to S3, write various types of metadata to a database, which in this case is Aurora Postgres instance in RTS. And finally, we terminate the EC2 instance, so we're not paying for idle instances when there are no jobs running. All these resources are located within dedicated VPCs and subnets to ensure security, and we make use of SQS to decouple the scheduling instance from the worker instance. So in this way, we are able to scale data processing to whatever extent we need and do it in a cost-effective way, so we're only paying when an analysis is running, and we're never paying for idle instances. Now, once the analysis is done, how does the user review the results in a manner that makes sense to him or her? So, as I mentioned before, the output is present in an HDF file, but this is a binary file and most people may not know what to do with it. So we want to provide a more visual representation of the contents of this file. So we came up with a run report that has a variety of key metrics and various diagnostic plots that help a user understand whether the experiment succeeded or failed. Now, this report is located in an S3 bucket that's been configured as a static website, and we use CloudFront as our CDN to enable quick delivery of these files to the user. And the user can view the report either within our application in the browser or download it and view it offline later on. And shown here are a simple set of plots that we show for a DNA plus protein run. Now, I won't go into the details of the plots. They can be very technical and require a lot of domain knowledge. But the idea here is that we have key metrics in tables. We have various plots that are currently interactive, and you're able to select subsets of data that you want to view and zoom into aspects of data. So this is a very user-friendly way of reviewing the results of single-cell data. And this matters because single-cell data is a new type of data. A lot of people are not used to it, and we are able to generate a visual representation of the results that enables a user who is new to single-cell technologies to understand what the data is telling them. This slide shows this very simplified reference architecture, and this highlights the use case where a user uploads a file via the browser. So the user goes to the browser, goes to the Tapestri pipeline site. This results in various API calls that have been implemented using API Gateway and Lambda. And as the user uploads files, the files are then put into S3 and then further processed by batch. When a user starts a job for the files that he or she has previously uploaded, this triggers the start of an EC2 instance via the scheduler, and all the communication between the scheduler and the worker happens via SQS. And the EC2 instance runs as long as it's needed till the job completes, and then it is terminated. But before termination, we copy data back to S3 and then write various information and details to Aurora. Shown here on the side are various supporting services that we use to enable this. IAM is used for security and access to data from a mission buyer perspective. So we restrict access only to those people who absolutely need access to customer data. KMS is used to encrypt data in S3, and this is mainly user data. Certificate Manager is used to provision our URLs using Route 53. Cognito is used to ensure that a user can only access their data and not some other user's data. And we use CloudWatch as a monitoring service so that we are notified about any issues that arise in the system, and we're able to monitor the performance of the system. So this was a brief summary of how the Tapestri pipeline web application was implemented using AWS. The various AWS services that are available enable us to provide easy ingestion of large FASTQ files, enable scalable and cost-effective data processing, and also enable us to provide a visual review of results that can be used to derive biological insights. And this is an example of how AWS is helping Mission Bio move precision medicine forward and help eradicate cancer. Thank you very much, and please visit missionbio.com to learn more about Mission Bio and the Tapestri platform.